Okay, good morning. Just give me a minute to check whether we are online or not. I'm not seeing myself here yet. Hello. Yeah. Am I audible? Hello. Hello. Yeah, okay. Looks visible. I think also looks audible. Let's start. Alright, so we will continue with histology of the male reproductive system. On a Saturday, I think it was 30th of May, I had taken the internal or the gonads, that is the testis, the epididymis and the vas deferens. I was told later that the PPT did not show, so at the end of this class today, I'll show you like a revision of those 3 slides. But I'm assuming that by now you've anyway finished the gross anatomy, the morphology, and at that time you would have also revised the histology alongside. There's no time to do all 6 tissues together. So today we'll continue with what happens after the vas. You remember that the germ cells were created, manufactured in the testis, and from there they moved into the epididymis, from the testis into the epididymis, and from the epididymis into the vas or the ductus deferens, and then they were brought to the ejaculatory duct which joined the prostatic urethra, where after which other things were added. But from here onwards, the seminal fluid is going to enter into the urethra and then be led to the external environment to be deposited in the vagina during the act of sexual intercourse or copulation. So this, whatever is being conducted in the vas, is production from testis and epididymis only, and epididymis doesn't add much. It modifies, it filters to some extent, but doesn't add much in terms of material to the semen, which mainly now is consisting of a whole bunch of spermatozoa. Once it arrives here, the addition of material from the seminal vesicles adds to the semen, and again once it arrives in the prostatic urethra, the addition of secretions of the prostate really builds up the bulk of the seminal fluid. The fluid part of the semen is added over here, and this is only done under sexual stimulation or arousal, and that is a very complicated, sophisticated, carefully orchestrated phenomenon where all these things happen together under control of the hypogastric plexuses. So there is a whole speciality called andrology, which deals with male reproductive function, just as gynecology deals with female reproductive function. So we won't go into that right now. We will just look at which part of that male genital tract adds what, and for the function of adding that, what does it look like under the microscope. So to revise, the gonads are a pair of testes in the male. The genital ducts include the epididymis, the ductus deferens, and the ejaculatory ducts. The accessory sex glands, apart from the gonad, they are accessory or helper to the gonads, are a pair of seminal vesicles which are situated here behind the bladder, a prostate, a singular midline prostate, a pair of bulbourethral glands which we saw in the deep perineal pouch. Then you have the male urethra, which is basically a passageway, and the penis, which is an external organ, this organ, in which the urethra runs, but which consists of other tissue also for the purpose of erection, so that the organ can perform intromission, that is entry into the vagina during sexual intercourse. So this also therefore becomes an accessory sex organ. It is more important for erection than for urination, but the urethra is the common passageway for both fluids, and it's either one or the other. So when urine is being passed through the urethra via the urinary bladder, we cannot have semen, and vice versa. So that coordination has to happen very nicely with the what shall we say, controlled entry of seminal fluid or of urine, depending on the function of the penis at any given one point of time. So let's get into the histology of it. So what we see is that the seminal vesicles are this pair of glands behind the bladder. The epithelial lining is of a pseudostratified tall columnar type and consists of secretory cells. And secretory cells are usually of a columnar type, as we have seen. In the cytoplasm, you have lipid droplets, giving it a foamy appearance. This we will see only under high power. I don't have a slide of that, but remember that it is pseudostratified tall columnar type of epithelium. The seminal vesicles produce this yellowish viscid alkaline fluid containing a whole bunch of molecules. They include fructose for the nourishment of the spermatozoa, some fibrinogen, vitamin C, and prostaglandins. The name prostaglandin indicates that it is produced by the prostate. You all must have done that in great detail in the class on prostate, so I won't go into that. But seminal vesicle secretion also contains a few prostaglandins. So the epithelium is, this is a very high power micrograph, is a pseudostratified. It looks like 2-3 layers, but they are not. There will be a few basal cells which do not reach the lumen, and there will be a whole lot of other columnar cells which reach the lumen, but there is a single layer of cells on the basement membrane and no double layers, right? So these epithelial cells contain also sometimes brown lipofuscin granules. You won't see them in HE staining, which have variable nuclear shape and size. That I think you can make out. They're looking more or less uniform nuclear size. Sometimes the nuclei are a little smaller, sometimes they are longitudinal, sometimes they are more flat, sometimes they are more square and basal. So variable nuclear shape and size. The vesicles are often seen to contain spermatozoa. This is the lumen which contains the secretion of the seminal vesicles. You must remember the passageway of the spermatozoa does not include seminal vesicles. The spermatozoa go into the ejaculatory duct. They are not supposed to come into the seminal vesicle, but sometimes, rarely, in a cut section you may see, because there may be a reflux from the ampulla of the ejaculatory duct by which it is going back into the seminal vesicle. A few spermatozoa may come back and be seen here. Not in this slide, but it can happen. So this here is basically the secretion of the seminal vesicle itself, and a simple or a pseudostratified columnar epithelium, no cilia, nothing. These secretions, it's a gland which is going to take these secretions and just empty them, join them to the ejaculatory duct. So this is what it looks like. Now inside this epithelium, it looks like there are pockets or like you had submucosal glands in earlier slides, but that's not it. This is basically a mucosa which is multiple folded upon itself. So there is a primary, secondary layer of folding of this mucosa. We saw this in fallopian tube, in the female genital tract. This is not that prominent. There are big lumens compared to what we saw in the fallopian tube, but in the seminal vesicle, we must remember it is a similar epithelium. Difference is fallopian tube is ciliated and this is not, right? And this looks very much pseudostratified. Fallopian tube is always simple columnar. In any case, you cannot confuse the two. You will see a muscularis wall which consists of an inner circular and an outer longitudinal layer supplied by sympathetic nerves, and during ejaculation this muscle contraction will empty the contents of the seminal vesicles into the ejaculatory duct, right, into the urethra via the ampulla of the ejaculatory duct. So this is the way the epithelium looks under very high power, and this is the secretions which have, this is an artifact, this diagonal look, that is the way the material, the solidified material has been how should we say, sliced by the microtome. But you only concentrate on the lining epithelium, pseudostratified tall columnar. Okay. This is what your diagram in the atlas looks like, a seminal vesicle with a low magnification. This is to show that there are two layers of muscle, the muscular coat having inner circular, outer longitudinal, and an adventitia outside, and what I talked about is crypts in the mucosa. That seems to be empty looking spaces or full looking spaces which are nothing but foldings of the mucosa upon itself. They are not separate glands, they are not mucosal glands, but they are crypts in the mucosa. Please remember that the depth of this, if it is cut like that, will look like that, right? So this is a continuous singular layer of lining. There is no ingrowing as a gland into, there is no submucosa with a glandular domain. So you have a glandular epithelium, glandular meaning epithelium of this gland, primary fold in the mucosa, secondary folds in the mucosa. Then you have the lamina propria and suddenly the muscle. So there is no submucosal domain as we have seen in other places. Mucosa, muscle, serosa. That's it. And in muscle there is inner circular and outer longitudinal. This is another diagram. So again, to show you primary mucosal folds, secondary mucosal folds, giving rise to the appearance of crypts. These crypts you see again in gallbladder. That is also simple columnar epithelium. But there you didn't have a submucosa. There also you had inner circular, outer longitudinal, or you had a more vague muscular domain. Just see if you can differentiate between gallbladder and semi-vesicle. But don't worry about it. The lining of the gallbladder doesn't look so very convoluted. The epithelium here is in secondary and tertiary folds, giving rise to complicated looking crypts. Comparatively, gallbladder is much simpler. And you have, okay, the other things are as told. Alright, so then let's come to the second organ. Here we had to looking at the penis as a gross structure to see that within the erectile organ called penis, there are actually 3 domains of tissue, which are called the corpora cavernosa and the corpus spongiosum. Spongiosum is placed ventrally, cavernosa placed dorsally, and they are paired. Spongiosum is single, unpaired. And inside it is the male urethra. Now this part of the urethra is the one after the bladder neck, which is a prostatic urethra, the deep perineal pouch, which goes to the urogenital diaphragm. So that's called the membranous urethra, and this is the penile urethra. So the third and last and longest part is the penile urethra, which is embedded within this round cylindrical tissue called the corpus spongiosum. And this is therefore urinary in nature. It has got nothing to do with the sexual function. The other two, the corpora cavernosa, are a what do they call that? A cavernous tissue, as the name suggests, which are capable of getting filled up with blood if the venous return is impeded. So if the venous return from the penis is impeded, these corpora cavernosa will fill up with retained blood, and that will make the corpora erect and stiff, and therefore will erect the organ itself. So the penile substance is made of these 3 masses, 2 dorsal corpora cavernosa and 1 ventral corpus spongiosum. The 2 extend right up to but not beyond the glans penis, whereas the spongiosum goes into the glans and the urethra within it opens at the tip of the penis. Here it is skin. The outside of the penis is skin. So the outside wrapping tissue is skin with all its layers, epidermis, dermis, and all the features. And therefore, at the urethral opening, there will be a stratified squamous, partly keratinized epithelium at the outer edge of the urinary meatus, because it is mimicking or blending in with the skin of the outer penis, outer layer of the penis, and the urethra otherwise should have had transitional epithelium, the urinary tract epithelium, as we've always seen, right? So remember that from the pelvis of the ureter onwards, the urinary tract always has the urothelium or transitional epithelium right up to the tip of the urethra, except where at the tip of the penis it becomes stratified squamous keratinized, matching and blending with the skin. Same thing happens with the anal bulge. The anal tract has an epithelium like the GIT, like the large intestine, but the anal exterior, after the anal sphincter, when you have the anal opening, again it will become stratified squamous keratinized. So where you have a mucocutaneous junction, you have the lips, you have the edges of the eyelids, you have the interior nares. They will be continuations of skin, and they will have the histological architecture of skin, because they are blending in and then becoming continuous after the whole oral oropharynx, then the GIT kind of lining will start. Because wherever there is going to be friction and attrition and drying up from the external surfaces, a stratified squamous is provided and a keratinization is provided on the dry parts of the surfaces. Okay, so coming back to our the second tissue which we look at of the penis, we know that this is what the structure of it is: an outer layer of skin with some fibroelastic tissue, a sheath-like thing containing two cylindrical bodies which in cut section look ovalish, which are called the corpora cavernosa, and one more cylindrical body, three of them together in a rough oval shape. So this is what they look like, two corpora cavernosa and one corpus spongiosum. But let's look at its features. So there is an arrangement of erectile tissues which are at the back or dorsally placed, and a single ventral collar which contains the penile urethra. And at the distal end, the corpus spongiosum alone expands to form the glans penis. These erectile corpora are enclosed and separated by a fibrocollagenous capsule. That means most of the fibers are of a collagenous nature. The surrounding of the corpus spongiosum also has its own thinner capsule which has a little more elastic tissue actually, whereas the cavernosa has more of a collagenous tissue. And the center of the penis is enclosed in a sheath of skin outside, which is connected by loose subcutis containing prominent blood vessels. So just under the skin you have a whole bunch of blood vessels. So S is actually subcutis. I've written it as skin here, but under the skin, the dark purplish layer is the skin. There is a subcutaneous domain, you can say, under which is this fibrocollagenous sheath. Inside also, this F is a fibrocollagenous domain. This is a fibrocollagenous domain. The erectile tissue is smaller than the dimension of the corpus, because the corpus consists of this fibrocollagenous layer, quite a bit of it, from this much to this much, and then the erectile part is in the center of that. So this is what it looks like at very low power. This is diagrammatic. I'll show you the labeling in the next slide. But basically this, if you got a low magnification of the entire structure of the penis, this is what it would look like. So you would have the skin, you would have the subcutaneous layer, subcutis, what they're calling, and then you would have this fibrous sheath, and then you'd have the individual sheaths, one for the spongiosum and the thicker ones, denser ones for the cavernosa. And even here, a lot of collagenous tissue, fibrocollagenous, and it contains some smooth muscle also, which is why it is so pink, whereas the smooth muscle of this one is much thinner, right? Thinner, lighter, whereas this is much thicker and heavier. And in the heart of it you have these blood vessels, including the deep arteries. So we will come to that. We had deep arteries of the penis. They are the ones which are going to be included here. So the corpus cavernosum is surrounded by a dense sheath. The sheath contains all types of fibrous tissue, collagen, elastin, and also smooth muscle fibers. In the midline, it is fused to form a median septum. This seems to be not separated, but there is a septum. They lie side by side, and the fibrous septum separates these two corpora cavernosa. The spongiosum is surrounded by a much thinner sheath and lies ventral to both of them. It lies in the midline. And an additional sheath, thinner one, surrounds all three of them together. That's what I said. So this is the general structure. So let's look at all of the labelings. So like I said, deep arteries of the penis, one in each corpus cavernosum. A dorsal artery should have been there. A dorsal vein is in the midline, deep dorsal vein, we saw that. Actually it should be called, okay. So this is underneath the fibrous sheath, and there is a more superficial vein which we see just under the skin. And then you have the deep arteries, the two dorsal arteries, left and right. And then you have the two deep arteries. You have nerves just under the skin. We've seen this in the gross anatomy of the penis, how it was lying. There was a vein in the midline, and there were two arteries, and then there were two nerves, and how they came under that arcuate ligament and got into the penis and became the dorsal structures of the penis. So we saw all of that. Then you have this tunica albuginea. I don't know if that is a correct terminology used in all textbooks, but since it is labeled here, we'll call it that. It consists of this fibrocollagenous material. You have a deep penile fascia. You have helicine or helical arteries inside of it, which will be corkscrew shaped. They're not arteries, they are branches of arteriole, so that's the wrong word. Even this, this you can call an artery, and it will be a big one because that's visible to the naked eye, but these will be arterioles. Then there'll be a little bit of a tunic of smooth muscle, and then there will be in the spongiosum, there will be the urethral tube, which looks like a very wavy, curvy lumen, and this will have the transitional epithelium. And on the outside, you will have epidermis and dermis, both of them together is forming the skin. So we've got everything here. You have a median septum where the two corpora are separated from each other. You have the cavernous tissue, which is essentially, all cavernous tissue is essentially a blood vessel type, right? So when you have vascular tissue, they have the original plan of tunica intima, media, externa. But here, because they are cavernous like sinuses, not capillaries but like sinuses, much wider space. The difference is the space available in the lumen is much wider and more full of blood, but the lining will again be simple squamous epithelium because of the blood content. So that's what each cavernous sinuses will look like. The trabeculae of little bit of reticular tissue in between, forming the bulk, the architect, the skeleton of the corpus cavernosum, and holding all these vessels and these collagen fibers and all together. Sebaceous glands, as you have in any skin domain, you've seen skin, right? So in that, and then you don't have hair, it's hairless skin, but otherwise it is normal skin which is covering the surface. So this is a complete separate organ domain, and then inside of that, this is the typical scaffolding available to keep the function of the penis possible. At all times, these corpora cavernosa are flaccid, they are not filled with blood. There is a normal blood flow, arterial flow, and venous drainage, and all that happening. Only during erection, you have a difference that firstly, this urethra won't be able to fill because there will be a constriction of the urethral sphincter. Then these two will be filling with blood, and the venous return will be impaired, will be shut down by autonomous mechanisms. Therefore, making these two areas distended with blood and stiffening the whole organ and allowing it to erect. Okay, the histology part I think we've covered. It's easy enough to see. Now we try to put it like this, a single view under a microscope, but then we'll have to put it under low magnification if we have it on a slide. But if you look at it under high magnification, we see that this is the look of the corpora cavernosa. A lot of vascular tissue, all this brownish red stuff is blood, and these are simple squamous epithelium lined cavernous tissue. And this is the connective tissue which is forming the fibrocollagenous surrounding, and which will form the septum between the two, which will send in some trabecular structures and some network like tissue holding it all together. Whereas this is to concentrate on the corpus spongiosum to show that this is also very well supplied with blood, good amount of vascular tissue, but this is not erectile in nature. And here you have the urethra, which is the main function of this part, which will have a transitional epithelium like you have in the bladder for most part of the urethra except at the tip. So this is the structure of the corpus spongiosum. Usually if you look at 3 bundles of tissue, we'll see this again in umbilical cord. You'll actually see 3 different vessels in it, and they are vessels, they are artery and vein. So you shouldn't confuse that with this. This is a completely different type of tissue. So look at the surrounding and look at the inside and look at what is there in the middle. The presence of a urethral structure in any one of these 3 is diagnostical. It can't be anything else. But sometimes, the minute you see 3 things, you think of umbilical cord. We'll do that, and at that time I'll again differentiate. If at low power we put these 2 organs together, they're very different, they stain very differently from each other. We will look at the differential points again when we do umbilical cord. But this is the penis, and particularly we're looking at the corpus cavernosum, which is the reproductive tissue within this organ. if you looked at just if I just wanted to show you a slide of urethra, right, then this, if I put this there, it would be urinary tract, that we're looking at the urinary system and not looking at the reproductive system at all. So then it would look like this. You'd have a kind of a you won't see a singular common small round oval lumen. It will also look somewhat star-shaped. This was the look in vas also, vas deferens, but that was more curved outline of the lumen of the vas, whereas this is more star-shaped. How do you describe it? Right? And then we look at the lining epithelium carefully. This one is transitional and that is not. So that is important to remember. And little bit of vascular tissue around, some amount of fibrous tissue surrounding it, a thin sheet of fibrous tissue surrounding it. And this is never standalone, it's part of the penis, because it's difficult to get a sample of urethra except from the penis. The prostatic urethra or membranous urethra is difficult to get it from the animal or anyways. And usually you don't see this in pathology unless a sample or unless a penile excision has been done for some reason. You're suspecting a carcinoma or you're suspecting some other pathology, some degenerative pathology, in which case the surgeon would have had to do a complete penile excision, and then we will study it under the microscope to see whether we got the entire malignant tissue and whether it is, what all has it infiltrated, therefore what lymphatic drainage might have been impacted, therefore how shall we stage this disease, therefore how shall we treat it. For that purpose we do a pathological biopsy. If we ever do that, and if you ever see it on a slide, then you remember next year when you see pathology slides that this is what a urethra is supposed to look like. It's not part of your undergraduate syllabus in anatomy histology, but this is what you remember where it is. It is part of the penis. Then let's come to the prostate gland. Now this is an important, very important gland and organ in terms of surgery, because lots of many middle-aged male patients will come to us with a condition called benign prostatic hypertrophy. You must have heard about that even in your gross anatomy classes. So we must remember the histology of it, because you will see diseases of it in pathology. So the prostate gland is composed of glands and stroma, and the supporting stroma is a mixture of, again, collagenous fibrous tissue and a little bit of smooth muscle fibers. The fact that there is smooth muscle in the supporting collagenous tissue differentiates the prostate from all other glands. We've seen so many glands so far. So none of them had smooth muscle in the stroma. So if you're seeing smooth muscle in stroma, then it must be prostate gland. Otherwise, all of them are tubulo-alveolar glands, isn't it? So compound racemose glands. So this becomes a differentiating feature. The glands themselves show a convoluted pattern. The epithelium is thrown into folds, almost like a papillary pattern. You just see it in the next slide. The secretory product, prostatic fluid, which makes up more than half of the seminal fluid volume, is a thin liquid rich in citric acid and proteolytic enzymes, including fibrinolysins. The purpose of this is to liquefy coagulated semen after it has been deposited in the vagina. Semen by its own nature will clump together and kind of become a jelly-like thing, in which case spermatozoa will not be able to swim upstream and enter into the uterine lumen and the fallopian tubes, where fertilization should take place. So the prostatic content is what allows the semen to remain liquid and flowing, and that's why these structures, fibrinolysins, proteolytic enzymes, are required for that. So prostate secretes it, adds it to the seminal fluid. The whole thing is added and deposited within seconds, within a minute. So the actual action happens in the vagina when the entire seminal fluid has been deposited there. Now the inspissated secretions, if the prostate has not been emptied in ejaculation over long periods of time, the secretions just sit there. They will only empty during the sexual act, not in between. So when you have retained fluid material with a lot of components in it, it can become concretized. It can get dehydrated while it is sitting there inside its own glandular lumen, and then it kind of condense upon itself and become a firm to hard small pellet, which then cannot be emptied anymore, because now it's become too solid to flow. So it remains back in the prostate. It's not of much significance, but the fact that these small sand-like particles remain behind in the prostate is one way to identify the organ and to differentiate this gland from other glands. For example, in extreme cases of dehydration, you can have salivary calculi also. The saliva which is not able to empty from the salivary glands becomes dehydrated and concretized, becomes like sand. And such a calculus can go and block the duct and cause a salivary obstruction. Rare, not very common condition, but it can happen with certain kinds of lifestyles or certain kinds of diet maybe, and certainly with dehydration in the patients. So, but that's a rare thing, and that is considered a pathology that has to be taken care of. Whereas a prostatic accumulation of inspissated secretion is a normal thing. It seems to happen more in older men. As age increases, the frequency of finding these secretions inside the prostate gland is more, and these concretions are called corpora amylacea, and they stain dense pink, so they're very easy to see. They increase in number with age, and they may become calcified. They usually don't cause any trouble to the patient. The patient won't have any symptoms related to this. It won't go and block the duct or anything like that. It just stays within the gland. But we have nominated it, we named it as a histological feature because it can happen. It can be shown in the slide and you may see it. Now what this part is, again a diagram. This is from the prostatic duct. This is the urethra within the prostate, and we are not looking at that today. We are looking at the remaining tissue, which is the glandular tissue. So you have a fibromuscular stroma. You can see pink smooth muscle in between the glandular tissue. This glandular parenchyma is of a simple type, tubuloalveolar glands, irregular alveoli with wide lumen, and cuboidal to columnar depending on the activity. So if it's in a high activity state, there will be columnar cells, and if it's in a resting state, there will be cuboidal cells, and the lumen will be full of something or the other. You can see one or two small things here. They may be found inside, and see inside the lumen you may find some things, whereas the other structures which are seen in cut section are arterioles and venules. Anyway, that is usual. Are we online? I think so. Hope so. Yeah. Okay. So you have the tubuloalveolar glands. These, similar to any glands anywhere, like we saw in the parotid, or we saw any of the salivary glands that we've seen before. What other glands have we seen? Pancreas, the exocrine pancreas. So it looks similar to that, but it doesn't stain so heavily. And the differentiating feature are two. One is that you have the columnar, simple columnar to cuboidal epithelium. Don't mistake this for pseudostratified, though that's what it looks like. Some areas may be pseudostratified, like seminal vesicle. You concentrate, for differentiation's sake, you concentrate on this stroma which has smooth muscle. This is not available anywhere else. So prostatic stroma, being fibromuscular in nature, is its differentiating feature. In some of the lumina of the glands, you will see some smooth pink staining, smooth material, which is called the corpora amylacea. And these concretions are more common as the male animal grows older. Okay. This is a much bigger picture. So you have a corpus amylacea and you have a lining epithelium, and you have, these are the secretions of the prostate itself. All kinds of material. They are not spermatozoa. They won't be seen within the glandular parenchyma. Please remember, spermatozoa may be seen here inside the prostatic urethra, because that is where the ejaculatory duct is entering. But that too, if you take a section of the prostate beyond the opening of the ejaculatory duct, not before, right? There is a length to the prostatic urethra. The upper half of it cannot show spermatozoa, whereas the lower half of it may show. But in any case, I have never seen in the prostatic slides. Whereas in seminal vesicle, because of reflux, some spermatozoa may go back and stay there, and that might get caught in the slice. So in the prostate, you're not going to be seeing spermatozoa anywhere. You may see corpora amylacea in the lumen of the gland, and the secretions of the prostate itself, including prostaglandins. This sometimes they ask you in the viva. You be prepared to know what are there are. I think some 8 to 10 molecules which are secreted, and all of them form the bulk, more than half of the seminal fluid, alongside the spermatozoa, which is live output from the testis, which has come from a long way. So this plus that together will then become semen and will get deposited to the outside. So this is the very high magnification. Again, please note smooth muscle in the stroma, in the areas separating the gland. You see pink smooth muscle, and that is prostate. That differentiates it from all the other kinds of glands that you see. So fibromuscular stroma with blood vessels, please call them capillaries, whatever else they may be. And this is the lining epithelium. It's drawn cuboidal here, but you saw it is more like columnar. And why is this? Sorry, this. Yeah, okay. Okay, I'm not seeing the bottom line, but it's okay. Nerves, blood vessels, lymphatics, and nerves. So it's supposed a parenchyma. Which one? The stroma. It's got connective tissue fibers in different directions and smooth muscle fibers. Remember that. This is a concretion or a corpus amylaceum. Plural will be corpora amylacea. They'll be very pink. They'll be smooth outline. They'll have a smoothish texture. Not really like this. You saw it's a very smooth texture, very homogeneous kind of a texture. And the other, this is the remaining prostatic secretions. So in a high power, that's what it looks like. Spherical or ovoid prostatic concretions, condensation of the secretions, and the number will increase with age. So revision, you have a seminal vesicle, you have the penis, you have the prostate, and in the earlier classes we had penis, epididymis, and vas deferens. Please revise all of these together in continuity if possible, so that then you get a sense of how the whole male reproductive system histology changes down the track. And later, when we get slides which are confusing, which are together, I'll try to show you juxtaposed with each other, so that how to differentiate, especially for spotters in your practicals. We'll try to do 1 or 2 sessions like that, which will help you to differentiate one kind of slide from another. Right? But these are not difficult slides. They're easy, comparatively easy. Not really confusable with the other slides. Okay, so I'll stop with today, and we'll continue with the remaining pelvic structures. I may be taking up, I think, the last of the female reproductive tract histology, and then if there is some leftover embryology or any other gross anatomy, I'll probably take it up. So have a nice day, and I'll see you on Saturday.